Hey guys, it's Mrs. Lloyd here. I'm coming at you out for Thursday, April 16th lesson, and we are going to talk about the action potential today. So, when we're talking about an action potential, all that is a nerve impulse. So, the electrical signal that gets sent down a neuron. So, anytime I use the word action potential today, think of it as the zap, okay? The signal being sent from your brain to your body. So, here in this picture, we've got a picture of a neuron. We've already gone over all these structures in your notes and identified these important structures. As you can see, this arrow on the bottom is showing you that direction of that action potential or the electrical impulse as it travels down a neuron. And all neurons are connected at their synapse. That was the lesson from previously this week. And then what happens after a neuron is stimulated is what we're discussing today. So, our main region that we're focusing on today is the axon and what is happening here. And action potentials can get very complicated. I've attached to your lesson today a few extra helpful videos I would encourage you to watch just to get the information in more than one way. But don't get too bogged down in all of the details. There's just some simple, basic things that I want you to know for your lesson today. So, on your notes, there's not an actual page for this next set. Just find a little section where there's a blank piece of paper. You're not going to need very much, maybe like 4 inches or something, to list out the steps of the action potential. So find somewhere to jot this information down. It's nothing that's previously in your packet already. So title this little section of notes, the action potential, and right next to it, write nerve impulse. So you know a nerve impulse is an action potential, right? This sending of a signal. If you recall, when we did the muscular system, we talked about membrane potentials. It's basically the same idea or concept of stimulating something else with seemingly electricity. Now, it's not electricity, it's actually ions. So I'm zoomed in here on the axon from the previous picture. And there's really just two main things to know. This slide has the first, and then the next slide has the second. So your membrane, when a membrane of a neuron, when it's not active, is at rest. And we describe that as a resting membrane potential. When it is at rest, we describe it as being polarized at negative 70 millivolts. Now, millivolts is just a way of registering an electrical current. That's not super important, but that number, negative 70, is when we look at our graph later. So write that down, the resting membrane potential. Okay, this is our first step, and then it's polarized at negative 70. Now, I want you to sketch down an axon. It doesn't have to look exactly like this. You could literally just draw a tube if you want to. But the important thing that I want you to do is add some charges. So a membrane at rest is charged or polarized. So something that's polarized means that it's got positive and negatives. And so this membrane has positives and negatives. All of the negatives are on the inside of that axon, and all the positive ions are on the outside. This is what the axon looks like when it's at rest, before a signal or nerve impulse is sent. Okay, this is what it is at rest. Lots of negative charges on the inside, lots of positive charges on the outside. And the reason why there's positives and negatives is because of the two main ions, sodium and potassium, and they are kind of what run the show here. One of the videos you'll watch, the first one, first optional video I posted, this is a screenshot from it, sort of showing what that looks like in more detail and explaining the resting membrane potential. So here we have a membrane. So this is the cell membrane. We got a phospholipid bilayer, throwback to biology. These are little gates that let ions in and out. You can see some come in one way, and then we got some others going another way in this picture here. And then there's that negative 70 millivolts. If you could measure the charge of this membrane, if you actually had a device that could fit into this teeny tiny space, it would register it at negative 70. The main thing I want you to know is that it is positive on the outside, negative on the inside when it is at rest. That's the important thing to know. So that's the first thing about an action potential is what is it like at rest? Negative 70 millivolts, positive outsides, negative insides. Okay. The second thing we're going to talk about today is how you generate an action potential. So how do you get it to go and move? So first step to know is that your resting potential is when it's polarized. That's what we just went over. That's step number 1 when we create an action potential. This all might sound very complicated as I go through it. Please just write these notes down and it'll make a little bit more sense here after this slide. So what has to happen in order for a signal to be sent is a threshold potential has to be met. So imagine that a mosquito landed on your arm and you didn't notice it. A signal was not sent to your brain because it was so light and fine that there was no signal sent. That would mean that the threshold potential was not met. But if you stubbed your toe and that painful effect was felt, that threshold was met, and that stimulus of stubbing your toe sent changes to the electrical gated ion channels. So the electrical gated ion channels are just those doorways that are going to let the ions in and out. And particularly, we're talking about sodium and potassium. All right. Then the third step is when the membrane becomes depolarized. So if it's polarized when the inside is negative and the outside is positive, to become depolarized means the inside becomes more positive and the outside becomes more negative. You'll see that on a diagram here in a bit. There's a period of transition, which we'll describe later, and then hyperpolarization occurs. So it undergoes this period of repolarization, and it ends up doing it too good. So the inside returns back to becoming negative, but it becomes overly negative. So the last step then is to try to even the whole thing out. So please list these different steps down in order. It will be important and helpful for your homework assignment. Now, this doesn't help you understand a ton about what's going on in the neuron. So I want to describe what's happening to those little gates. Those gates are what are letting in and out all of those ions, our sodium and potassium, that causes this change in the positive and negatives. So at step number 1, when your neuron is at rest, the gates are closed. Okay. So these things in parentheses here are what are the gates doing? So when you reach the threshold potential and you're going to send a stimulus, these sodium gates are the ones that open and a bunch of sodium rushes in. And this is what ends up making the inside a little less negative. Now, as it is becoming depolarized, these sodium ion gates end up closing. We end up having this transition period where the potassium gates are opening. When it is time to go back to hyperpolarization, those sodium ions are very slow to close, and this is why it hyperpolarizes. And then eventually you get back to rest. And that's thanks to the action of this thing called the sodium potassium pump. So on this list of notes here, Na is sodium and K is potassium. So we describe these as sodium potassium pumps. Now, I do know that both of these ions are marked as being positive. So you might be confused about how it becomes negative if positive ions are the ones going in. It gets complicated. Just associate the potassium sort of with the negative charge. It's not so much that it's negative, it's just more negative than the sodium positives. Okay. This picture, I hope, is a more visual of showing what's happening with these gates, opening and closing. So we're going to be labeling this picture here elsewhere in our notes here in just a second. But here you can see negative 70 millivolts. This is what your membrane is like at rest. And then if I meet my little threshold, okay, a mosquito landed on me and it was strong of enough force that I noticed it. I needed to hit at least negative 55 millivolts in order to reach threshold. Then a signal is going to be sent. When that signal is getting sent, we say that those sodium channels are open, okay? Right here, that's this step right here. And so all those sodium channels are open. At the tippity top here, this is kind of our transition where both different types of channels are open. Right? We have potassium ions right here that are opening. The sodium channels are closing. So then we go back down because it's becoming more negative on the inside. When I talk about hyperpolarization, it's this little section right here. Notice how it dipped below negative 70? That's because it became hyperpolarized. And then eventually we get back to our normal repolarization at negative 70. So this is what it looks like, okay? So here is my neuron, okay? This picture is maybe actually a little bit better. Here's my neuron and my action potential, my nerve impulse is getting sent. When it's at rest, it's negative on the inside, positive on the outside. The main thing I want you to know is when an action potential is being sent, that charge flip-flops. So right here, where it's positive on the inside and negative on the outside, that is the action potential being sent down. This section of the neuron hasn't been affected yet, but you can assume that these charges are going to flip. Now, it goes back to normal, right, when it depolarizes. And so this has already gone through that phase of depolarization. The videos I've attached are helpful because it shows all this in action instead of just a 2D picture. Now, myelin sheaths made by the Schwann cells are super important because they make the signal get sent faster. What ends up happening is you see these little arrows here. The action potential kind of jumps in between each of those nodes of Ranvier that we labeled before. And so when the action potential is occurring, okay, it ends up happening more quickly because it's skipping over those little sections of insulation and we're able to send signals more quickly. So myelinated axons, anytime we have a myelin sheath, they send the impulse way more quickly than unmyelinated axons like this one. All right, so these are two more pictures. This is another little snapshot. I think this is the third video showing you the effects of the myelin. What I like about this video when you watch it is each one of these little up and down is showing you one picture of that graph that I showed you where it's depolarizing and repolarizing. And so it happens at every single one of these little nodes here. And so it's like a blip, 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 blip as it moves down our axon. That video is good to watch. What's important to know is that the myelin sheath speeds up the reaction. But I like these two pictures here, okay? It's showing you that the Action potential is getting sent. Okay, in this case, it's going from right to left, this direction, right? Because this is the direction of the propagation. But what's happening here is this is the membrane at rest, negative inside, positive outside, and the signal, as it moves down, those charges flip-flop. That's the really important thing to know. As we send an action potential, the negative inside becomes more positive as it moves down. All right, so this is a page in your notes close to the back. It is on page 10 in your study guide packet that I provided for you. So I want you to fill this little tree chart out, and we're going to reiterate some of these important things. So our nerve impulse, first thing to know is, what is it like at rest? Well, when it's at rest, we describe it as being polarized, meaning it has a positive outside, negative inside. That is what polarized means. You might draw a real quick sketch, just show a little tube, put some pluses on the outside, some negatives on the inside to show that's what it looks like at rest. If we were to measure it, it would be negative 70 millivolts, and it gets maintained by this thing called the sodium-potassium pump. The video will show you more detail about the sodium-potassium pump. It's not super important for your test that you have all these steps memorized, but the sodium-potassium pump is sort of what maintains this entire membrane and keeps it at rest, so the inside stays more negative and the outside stays more positive. Now, when I'm ready to send a signal, I go through a bunch of these different steps. And this diagram right here, we've got these different steps here labeled. And so I want us to record the numbers down about what's happening at each of these. So where it says number 1 on your graph, that's the resting state. So make sure and add a number 1 so you can associate that with it. That's when it's at rest. That's our resting potential when the membrane will be polarized, negative 70 millivolts. In order to reach, or send the signal, the first thing that has to be met is the threshold potential. There's no number here, but you can see right here where it says negative 55. Right there on the graph is where the threshold potential would be. Many graphs show a dotted line across here, so you can kind of see where that threshold is. And if the line goes above that threshold, it means we are gonna send a signal. Step 2 on the graph is this section of depolarization, where the membrane is becoming depolarized. So the inside's becoming more positive, the outside's becoming more negative. At the tippity top, number 3, is this transition point, which I don't have listed here on the notes. But then as the roller coaster drops back down here, number 4, so make sure and write 4, not 3. This is where we describe it as becoming repolarized, going back to normal, meaning negative inside, positive outside. Then your body does too good of a job and it goes past negative 70. So it does what we call as hyperpolarizes. So it becomes overly negative on the inside, and we end up having a hyperpolarized membrane. So then finally, the last step would be occurring right here, where it's going to return to its resting state thanks to those sodium-potassium pumps that maintain it. So I like this picture a little bit better. It shows you what's happening at each of those steps. The only number we don't have labeled here is number 3. That's just the transition point where some gates are closing and other gates are opening, etc. All right. And then the last thing is, how fast do these signals get sent? Well, thanks to the myelin sheath, you are able to speed this up very, very quickly. Another fancy word for speed is propagation. So they describe this, and you'll hear this word on the videos, but they describe it as a saltatory conduction. That word just means to leap. So it's that idea that when there's myelin sheaths on there, it jumps over the little nodes and this ends up speeding up the conduction. So the myelin sheath is responsible for making this happen more quickly. So most of your nerves, especially in your peripheral nervous system, are going to be myelinated, so they send signals more quickly. This is a really difficult topic, you guys. And so I listed 3 optional videos in Plan Book for you to watch. Many of them I referred to in here. They're all 3 pretty short. And then there's one other that I said, do watch this one because I think he does a good job with his graphics of showing you all of these different things. Don't feel bogged down if this doesn't make a ton of sense to you. Hopefully you can do your homework assignment from this basic information. That's the main goal. Don't feel like you need to memorize every single one of these little steps though. Thank you guys. I hope you have a great Thursday. Bye-bye.